we've got Robert Peach up next. Robert is on the board of AdAlta. He's been working in the drug development space for 25 years or more. He's the co-founder of Receptor. So Receptor, he was the chief scientific officer and founder. They raised $800 million in an IPO over a 12-month period, and in August 2015 was sold to Celgene for US $7.8 billion. So he's had significant experience developing drugs, so not only the drug that went into receptors, but before that at BMS. And the drug is very similar in format to AdAlta's AD214, so he'll give us a bit of an overview of that. But originally a New Zealand boy, so we won't hold that against him.
So is this a hostile audience? Is it? All right, so Sam asked me to give this audience a little overview on therapeutic FC fusion proteins. The reason for that is because of the format of the lead asset that is with AdAlta. It is a therapeutic FC fusion protein, and they're not that common, but I think this is an opportunity to pretty much educate people and let people know what the format of this type of drug is and why it's as advantageous as it is. So I'll describe the nature of FC fusion protein, and then I'll argue about why we utilize the FC construct as a therapeutic modality. And just as a spoiler, it's all around extending the half-life of a drug and being able to dial in and out certain effector functions that could be particularly relevant, especially in cancers. And there's a big issue that is around ease of manufacture, and I'll let Dallas talk a lot more about that. But that's also a key feature of therapeutic fusion proteins. And then I'll go into some sordid personal experience I've had with FC fusion proteins back when I first went to the United States in 1991 and worked for Bristol Myers Squibb up in Seattle. And I had the great fortune to work on a couple of really good projects that ultimately led to drugs that are now registered, and those drugs are called abatacept and belatacept. And then our final slide on some of the marketed therapeutic FC fusion proteins, and I'm sure all of you will recognize some of them. So a little bit of Immunology 101 here. This is a structure of a typical IgG antibody. So an IgG antibody is made up of two identical so-called heavy chains, that are these two chains of polypeptides here, and two identical light chains. This region here is called the constant region, or the FC region, and this region up here is where the magic happens, where antibodies bind to antigens, and you get this marvelous diversity in immunology, where pretty much every antibody has slightly different specificities, so that they can counteract any invading pathogen or whatever. These heavy chains are held, these light chains are held to heavy chains through a chemical bond called a disulfide bond, and as well the two heavy chains are held together by disulfide bonds. And what I'll be focusing on is this region here, the hinge, constant heavy 2 and constant heavy 3 regions. So what is a FC fusion protein? So this example is a typical receptor that you might be expressed on the surface of a cell, where the N-terminal of the protein is expressed on the outside of the cell. You've got a transmembrane domain, and you've got an intracellular region that's typically the C-terminal of the protein. This is a stylized view of the human IgG antibody that I just showed you prior to this. And this is what a fusion protein looks like. So what happens is you take the cDNA that encodes this region here, and genetically fuse it to the cDNA encoding this region, for instance, in this case of a type 1 receptor. And that cDNA construct is then transfected into cell lines, transcribed and translated, and the resulting protein is released out into the culture media of the cell cultures. So this is what we're talking about when we talk about an FC fusion protein. There's some interesting advantages not only around this FC region, but you have a dimeric structure here. And so 8214, for instance, well, I'll show you in a moment, is also the I-body is attached here, and you end up with a dimeric construct that gives you something called avidity, and avidity is very important because it strengthens the binding between this area and the target that it's going after. So avidity is an extremely important thing with fusion proteins. So here's the stylized look at an FC fusion protein. This region here can be the extracellular region of a receptor. It can be a cytokine, it can be an enzyme, it can be a peptide, and in AdAlta's case, it's an I-body. So this is the region that provides the specificity, if you like, for the target that you're going after. This region of a typical antibody, the FC region, provides certain effector functions called antibody-dependent cellular cytotoxicity or complement-dependent cytotoxicity. But also, very importantly, this region binds to something called the Neonatal FC receptor, which we all have, and which extends the half-life of this particular reagent. Extending the half-life is important because if you're trying to inhibit a interaction in a meaningful and clinically manner, you want to have your drug around for as long as possible and in circulation. And so extending the half-life, keeping it around to enhance inhibition of what you're trying to affect is extremely important. So this is what 8114 looked like, the original lead product that AdAlta had. And now what we've got, the lead program is 8214. So here's the I-body. Here's that same I-body, but this time there's two of them, and they're attached directly to the FC region of an antibody. So that's the big difference between 114 and 214. And this structure imparts all of these very important properties to the drug that we believe are beneficial over the 8114. So just to get into to briefly describe what happens in terms of extending the half-life. So antibodies cross the maternal fetal blood barrier, and they do it through a receptor called the Neonatal FC receptor. So you see that in the placenta. Mother's antibodies get transferred to the baby's circulation via this mechanism. But also this neonatal FC receptor is expressed on endothelial cells throughout the body, and it is there to impart and provide a longer half-life for circulating antibodies and for albumin as well. And what happens is there's this pinocytotic activity where there's a non-specific uptake inside a cell. Antibodies that are taken up bind to the neonatal FC receptor, and then they get recycled back to the surface. If they don't bind, or there's an inability to bind to the neonatal FC receptor, they go through a lysosomal degradation process. So this whole process here is what the basis of an extensive long half-life is provided by the neonatal FC receptor. So typical antibodies in circulation have a half-life of around 3 weeks. You compare that to, say, a small molecule drug that maybe 24 hours or even less sometimes. So these protein biologics have quite extended half-lives. Speak up? Okay. The other part of the FC region that's important beyond extending the half-life of a drug is these so-called effector functions. I won't go into it in great detail because it is quite quite complex, but you either get, you can get complement-mediated cellular cytotoxicity, or by recruitment of macrophages and natural killer cells, you can get an antibody-dependent cell-mediated cytotoxicity. This is particularly important if you want to develop an FC fusion protein, say for a cancer indication where you want to kill a target cancer cell. So a good example would be rituximab, an antibody that I worked on in San Diego some years ago with IDEC Pharmaceuticals. Rituxan binds to CD20 expressed on the surface of malignant B cells. When the antibody binds, it recruits in these killer cells, macrophages and natural killer cells, and also activates complement pathways that lead to cell killing. So you can dial these effector functions in and out, which makes it a very robust platform approach to drug development. In the case of AdAlta, AdAlta's not, the drug's not in the business of killing cells, and so these kinds of effector functions have been dialed out of the construct so that we're not inducing cell killing. Now, I just want to spend just a few minutes now talking about some of the experience I've had with therapeutic FC fusion proteins, and it does date back to 1991 when I first moved to Seattle, and we're working on developing immunosuppressive drugs to block lymphocyte activation. Now, in those days, it was understood that in order for an effective immune response to occur, a T lymphocyte needed to get two signals from an antigen-presenting cell. One of them was related to the peptide antigen, so whatever's driving the immune system, binding to a particular T cell receptor that's expressed on T lymphocytes, and that's called signal 1. Now, that alone doesn't activate a T cell to mount an effective immune response against a virus or whatever. You need a second signal, and it came through a receptor called CD28. So CD28 binds to two different cousins that are expressed on antigen-presenting cells called CD80 and CD86. When either of those receptors binds to this receptor, in combination with that interaction, you get an effective activation of the T cell and a full-blown immune response. It turned out that there was another receptor called CTLA4, which is a very close homologue to CD28. Now, it was cloned by a French group in the late 80s, and because of its homology to CD28, and because CD28 was known to be very important in immune responses, we spent a lot of time studying CTLA4 to see what its biological function was, and see if we could hijack that biological function to develop a drug that would be immunosuppressive. So what we did is we made a soluble form of the CTLA4 receptor by hooking up this extracellular region of the protein to the hinge and constant regions of an antibody, and made this FC fusion protein. I'll just add that this was an extremely productive area of science at the time, and CTLA4 turns out, I mean, Yervoy is an anti-CTLA4 antibody that was the first antibody, so-called checkpoint inhibitor, that boosted immune responses against tumors, and is a very, very successful drug. The guy that originally developed that, Jim Allison, just received a Nobel Prize for his work around in this space. So this is what the CTLA4AG, as we called it at the time, what the fusion construct looked like. So you've got the full extracellular domain of CTLA4 hooked up to the hinge and two constant regions of a human IgG1 antibody. We had a disulfide bond here that existed between these two domains, so it came out as a homodimer. So we had this high avidity product that bound with very high affinity to its target receptor. We introduced mutations in the hinge region at the time so that we could prevent effector functions, this cell-mediated killing effects. And the reason for that is we didn't want to kill cells. And what happens is CTLA4AG comes in and is able to bind these two receptors with very, very high avidity binding, and it prevents those two receptors from interacting with CD28, so you shut down the immune response. You get a very potent immunosuppressive immune response. We tested this, we started off with this making this construct as a research tool, actually. We wanted to try and understand the receptor biology around these receptors and how we could potentially manipulate them. And ultimately, this therapeutic protein turned into a drug. So it's now called abatacept, and it's marketed as Orencia. Its half-life is approximately 14 days in humans, which is a nice long half-life, around 5 to 7 days in monkeys. So this ability to bind to neonatal FC receptors is working. And it's indicated to treat moderate to severe rheumatoid arthritis. It was indicated, approved in the USA in 2005, in Europe in 2007, and then Bristol Myers Squibb extended the activity of this drug in some nice life cycle management activities, expanding it into juvenile idiopathic arthritis in 2008 in the US and 2010 in Europe. Last year, worldwide sales of that drug was approximately $2.6 billion. So it's been a very effective immunosuppressive drug, been very effective and competitive with the anti-TNFs for treating rheumatoid arthritis. But there's a lot of life cycle management going on with this drug, and I just did a quick search recently on clinicaltrials.gov, and these are the ongoing Orencia clinical trials around the world. So these are all predominantly autoimmune diseases, and you can see there's a very wide range of diseases where this drug might have activity. The other one that I worked on, and this was something that was near and dear to me, was we had CTLA-4AG, and we understood a lot of the biology of what was going on in this synapse, this interface between antigen-presenting cells and T lymphocytes. CTLA-4AG was very effective, most effective at blocking the CD80, and could still block CD86, but not as effectively. And we thought a lot about this, and we thought about this in the context of solid organ transplant rejection. And it turns out that this molecule, CD86, is one of the first receptors that's engaged when a patient has, for instance, a kidney transplant, and a very potent immune response kicks off against the grafted kidney, for instance. So what we did is we did some mutagenesis work where we changed some amino acids in a couple of these loops of CTLA-4. These are loops that actually remarkably look a lot like antibody loops. We changed a couple of residues, and that increased the binding avidity of this mutant molecule, which we called LEA29Y, significantly towards CD86, and maintained activity against CD80. And then we thought, we've got ourselves a new drug. So this was a mutated variant of CTLA-4AG, which we took advantage of and then took it into a transplant setting. I had a collaboration with some transplant surgeons at Emory University in Atlanta. They were fantastic guys, and we did some, just as an aside, it took me 6 months to convince my company to give me the $500,000 needed to do the kidney transplant experiments. My boss wouldn't give me the money, his boss wouldn't give me the money, and then I had a fortuitous lunch one day in the cafeteria at Bristol Myers Squibb, where I happened to sit beside the administrative assistant to the head of R&D, and she said, "Do you want me to get you on his calendar?" I said, "Hell yeah!" And so I showed up at his office one day. He didn't know who I was, and he said, "What do you want?" And I told him, and we went through it. And 20 minutes later, he said, "How much do you need?" I said, $500,000. And he said, "You got it." So, yeah, I was persistent. I mean, we might be talking about this later on, but about what makes a drug, and one of them is persistence. So, yeah, so I got skinned over that one, but anyway. So this is monkeys given kidney transplants from other monkeys, so an allogeneic transplant. If you treat them only with CTLA-4AG, the animals reject the kidneys very quickly, and that's through measuring serum creatinine levels, so a surrogate for kidney function. You treat them with LEA29Y, and as a single agent, and you get much more prolonged kidney survival. We'd never seen this before, and the transplant surgeons were amazed. Mycophenolate mofetil and prednisone are part of a cocktail of drugs that are used for patients that have transplants, and they don't afford much protection just on their own. But when you combine the LEA29Y with these two drugs, you can see a very prolonged graft survival, which again was it was this experiment that convinced our company that we should advance this program into clinical development and ultimately use it as an agent for treating solid organ transplant rejection. And it's now called Neonatal. It's marketed in the USA and Europe. It got launched in 2011. It's a drug that has a half-life of around 8 to 10 days, a little bit less than Orencia, and around 5.6 to 9 days in monkeys. This is a steady state half-life, not a single dose half-life, a steady state one. So they are the two drugs that I spent through the 90s is when I spent a lot of time on that. But this is a list of all of the marketed FC fusion proteins that are on the market today. And this, of course, doesn't account for the numerous FC fusion proteins that are in clinical development. But you should recognize some of these. The first one that actually reached the market was Enbrel. That was developed by scientists at Immunogenicity in Seattle. Great scientists at real groundbreaking stuff. We were actually competing pretty heavily with them. They beat us to market. We thought we'd get to market before them, but we had some company politics that slowed things down, and we came in after Enbrel. But nonetheless, Enbrel's a huge drug. 2017 sales of over $8 billion. Orencia, $2.6 billion. Eylea, you might recognize Eylea for treating wet AMD. That's a $6.3 billion drug in annual sales. And then Trulicity is the latest one, a GLP-1 peptide analog for treating type 2 diabetes. That was most recently launched in 2014, and that's a $2 billion drug. So the bottom line here is this is this format for the types of drugs is a very robust format. It's very flexible. You can dial in and out various functions depending on which disease you want to go after. It affords a much longer half-life. And for me, it's and I think for the AdAlta team, it's a great format for developing these eye bodies. I think based on everything, certainly the experience I've had on it, I think an eye body as part of an FC fusion protein is absolutely the way to go to develop this drug. So, any questions?
What are the downfalls of using FC fusion with eye bodies? Are there any sort of close test cases of using eye bodies?
Well, so eye bodies are new, right? And we're learning as we go. I'm sure you'll hear from Dallas about manufacturing of these eye body FC fusion proteins. I mean, when you make an FC fusion protein, it's a novel protein. It's new, with the eye body hooked up to the FC region. There's a potential for immunogenicity there. So when you treat patients, they might mount immune responses against something that's novel or unique. But we certainly didn't see that with either Orencia or Neonatal. But that's a formal possibility. The other is just the behavior of the proteins. These are very stable protein folds in general, and so it's quite remarkable that you can hook these essentially two different proteins up and retain full biological activity. But that's the case. But I remember some of the fusion proteins were made, for instance, aggregated significantly. We never could truly understand why they aggregated, and aggregation can be an issue with novel proteins like this. But you know, it's a matter of doing the science and understanding what you've got and fine-tuning it where necessary, and appropriate formulation, and you've got yourself a drug.
Are these all injectables, or are they oral?
They're all either IV injected or subcutaneously injected. Yeah.
Or in the eye.
Or directly into the eye, in the case of Eylea. Yeah, there's so proteins. You essentially can't deliver proteins orally. I mean, your gut is designed to break down proteins, and so by the time a protein got, it just wouldn't survive the digestive system. So it's either intravenous or subcutaneous, and most of them are going for subcutaneous administration for convenience purposes. There are limitations to subcutaneous administration. You can only administer so much volume at a time, and if you limit the volume, you've got to have high concentrations of protein. Sometimes at high concentrations, proteins do funny things like aggregate and cause problems.
Rob, can making the protein larger, can that have a detrimental effect on the efficacy if it's not as nimble?
No, I don't think so. I mean, the key is when you make that fusion protein, confirming that you've got the retained biological activity, and that's what we've got with AD214. And in fact, I expect the activity of 214 to actually be superior to 114 because it exists in a dimeric state. So you've got instead of having one eye body, you've got two eye bodies essentially, and I think that's very advantageous in the context of disrupting, for instance, the CXCR4 and the ligand interaction.